chairwoman from the chairwoman from the morning session, Matilde Lleonart from Vall Research Institute in Barcelona, Spain, and she will tell us something about the syntenine overexpression and its impact on chemoresistance in cancer. So Matilde, the screen is yours, and we are listening.
Thank you very much for inviting me, for these participants, and especially for VHIR. So I will, in a second, to share the screen. I believe I have the same problem for sharing the screen. Okay, I will start talking about syntenine and how it modulates stemness and chemoresistance in head and neck squamous cell carcinoma. Head and neck squamous cell carcinoma is the 6th most common cancer around the world. And it has a local recurrence in the cervical lymph nodes. And in most cases, in the moment of the diagnosis, there is a second metastasis. So it's quite an aggressive cancer type. So here is in the whole panel of all cancer types, this is the mortality and incidence of different types of head and neck squamous cell carcinoma, including larynx, different pharynx, and also comprise mouth and nose. In Europe, we have the highest incidence and also mortality of the most frequent types of head and neck carcinoma that are pharyngeal and laryngeal. Between the risk factors, it's associated highly to smoking, to alcohol, and also to the infections for the papillomavirus. And even if there is a panel of genes that are involved in this cancer type, unfortunately, the therapy has hardly increased in the last 40-50 years. So the only regimens are cisplatin, and apart from surgery, and also radiotherapy. So there is a great need for find some kind of therapy for this aggressive cancer type. When it's detected in the early stage, the success of overcome the disease is quite high. But as I was saying, that in most cases, there is a second focus of the metastatic tumor, and then the diagnosis is quite poor. In our laboratory, we perform the generation of different resistant cell lines by intermittent exposures to different drugs. We focus on the treatment, especially in cisplatin and 5-fluorouracil, that are the most common treatment in head and neck cancer. So after a long period of time, we obtain resistant cell lines. We characterize genotypically and phenotypically in our laboratory, and also we generated the cancer stem cells from the parental cells of these 4 different types of head and neck cell lines. We check that the resistant cell lines are quietly associated to the acquisition of stem cell characteristics, and they also overexpress a high proportion of stem cell genes. On the other hand, when we started this project in the laboratory, there was not available cell lines from the pharyngeal, even in the ATCC or the European branch. So we generated from the patients from our hospital, we generated a collection up to 40 different primary cell lines from laryngeal cancer biopsies. So then we decide to inquire which proteins are involved in resistance. For this purpose, we start from a established cell line, the CCL138, that is a metastatic cell line from pharyngeal cancer. And we did an exhaustive proteome study from the parental cells the cancer stem cells and the resistant cells that were generated at our laboratory. So the proteomic study provides us a total of 36 dysregulated proteins, 12 upregulated and 24 downregulated. Among these 12 upregulated, we focus in synthenin 1 or SDCBP. First, we wanted to check if the proteomic data were correlated by other technique like Western blot. And we check indeed that for the CCL 138 cell line, in the resistant and also in the cancer stem cell derivatives, the synthenin was overexpressed. And also this was confirmed by other cell lines like HTB43, JHU029 and SSC25. So from all the panel of these 4 head and neck cell lines, synthenin 1 was more expressed in the CCL 138 and also in this other one that is derived from the laryngeal. In parallel, we designed a spontaneous model of cisplatin resistant. For this purpose, we injected HTB43 cells or CCL 138 cells into the mice, and then we treated them with cisplatin, so versus the untreated group. So in the untreated group, this is the black line, we see that the tumor grow, but in the treated with cisplatin, the effect of the drug was considerably, but then a proportion of tumors were at the end of the day overcoming this resistance and they were growing again. So after the experiment, we sacrificed the animal and we explanted the tumors and we put the cell lines in culture. And when we determined which cell lines derived from those mice tumors were resistant or sensitive. So accordingly, with the IC50 values, we assign or well sensitive or resistant cell line, and we observed that in the resistant cell lines, synthenin was overexpressed in the case of the HTB43 or in the case of the CCL 138. So then we confirmed that synthenin 1 is involved also in the acquisition of cisplatin resistant, not only in vitro, but also in vivo. Next, we wanted to check if the inhibition of synthenin compromised resistance in these 4 established cell lines from head and neck cancer. So we observe with CIRNA experiments a strong inhibition in all cases of synthenin, and the reduction of this expression, the reduction of the response to the drug, to the cisplatin. So in all cases, we obtain for the 4 cell lines, we obtain a reduction from a range to 10 to 35%, accordingly to the EC50 values. Next, we wish to test the biopsy-derived cell lines, and we observe that the inhibition of synthenin, this is 3 primary cultures of 3 independent patients with different degrees of resistance. In all cases, the inhibition of synthenin, we can see here the reduction values, was even stronger than those observed for the cell lines, ranging from 47% to 67%. We verified the inhibition of synthenin by an independent CIRNA, and we observed that our results were confirmed. So in all cases, we observe a reduction, for example, for the HTB43 resistant cell line, a reduction around 20%. This reduction was also correlated with a decrease in the number of spheres per well in both cell lines. So somehow seems that synthenin was compromising the cancer stem cell phenotype. The inhibition of synthenin was maintained quite long, over time with CIRNAS. And in the case of the CCL 138 or 43 was maintained till 9 days, and for the laryngeal cell line till more than 15 days. So we observed that indeed, when synthenin was inhibited, the spheres, the size of the spheres in 3D cultures was smaller, and also the number was compromised. In parallel, we analyze in the condition of the parental cells versus resistant cells, the inhibition of synthenin, and analyze all different panel of genes in relation with stemmen, including NANOX, SOX2, OX34, CD133, CD44, ABCB1, or KLF4. So we observed that in most cases, the ABCB1 or CD44 were decreased in the case where the synthenin was inhibited. Here we can observe that we corroborate the pictures from the previous slide. When in the conditions when synthenin was inhibited, was a decrease in the spheroid area and the number of spheroids per well. We analyze indirectly the cancer stem cells by the site population method by flow cytometry, and we observe that in the case of, accordingly with this table, in all cases where synthenin was inhibited, was a reduction in the proportion of the site population in comparison with the parental cells. So indeed, the inhibition of synthenin was decreased, not only the cancer stem cell properties, but the number of cancer stem cells themselves. We wish to analyze the ABCB1 protein, as we found it that was inhibited by mRNA in those cells where synthenin was inhibited. And we corroborate by another method that indeed, when synthenin was inhibited, we observe a reduction in this drug-resistant marker. We can check in the table the reduction percentages in those cases of parental or resistant cells where synthenin was inhibited. In contrast, in parallel, we overexpress synthenin just to check the increase, the reverse phenotype, and in this case, the increase in the ABCB1 marker, and we observed that was indeed an increase in this marker. On the other hand, synthenin depletion was responsible for the induction of apoptosis. So we observed that early and late apoptosis both was induced in different cell lines of head and neck upon the inhibition of synthenin. So indeed, synthenin was compromising the proliferative ability of these cells. We want to check as well, apart from the mRNA data, if by protein, we were verifying a change in the potential markers of related with stemness, including SOX2 and CD44. So even if SOX2 was not in all cases inhibited clearly, we observe a clear and strong reduction in CD44 marker for all cases. In parallel, we analyzed another protein that previously in the laboratory we had evidence that was potentially involved in resistance, and we analyzed the status of the phosphosarc kinase. So we observed that again the phosphosarc kinase was quite inhibited. The phosphorylation of this kinase was inhibited when synthenin was depleted. As SARC, we have the hypothesis in the laboratory that SARC may have a key role in resistance. We decide to inhibit SARC to see if somehow we could obtain similar results that inhibiting synthenin. So the inhibition of SARC, this is by CIRNA, so we were expecting to inhibit phosphosarc and total SARC. That was the case for all cell lines. The inhibition of SARC was compromising, again, the CD44 protein in most cases. The inhibition of SARC as well was provoking the same effect of syntenins, because it was sensitizing the cells to the effects of the cisplatin. Moreover, the inhibition of SARC, as we can appreciate in this table, in the bottom part, were provoking a nearly 50% reduction of the ABCB1 resistant marker. So in order to corroborate the results of the inhibition of syntenin by the opposite effect, by overexpressing syntenin, when syntenin was overexpressed by different plasmid, in this case we use the pcDNA, standard plasmid, we observed that the increased level of phosphosarc were significant in comparison with the control cells. We observe as well an increase in the CD44 marker, an effect more pronounced in the CCL 138-6, and also in this cell line an increase of SOX2. So when we analyze the IC50 values of the HTV43 and the CCL 138 cell lines, we observe that was an increase in the resistance. So indeed, the role of syntenin was quite specific in the mechanisms of resistance. This is the expression of syntenin by another plasmid, the IRES and GFP. We could monitor the cells overexpressing consistently by the fluorescent microscope. So we can check by mRNA levels that our vectors was specific and was increasing the expression of syntenin. And also we observe overexpression of phosphosarc, and at the longer time, an increase in CD44. So when we analyze again, by mRNA, the different stem cell genes, including ABCB1 and CD44, we observe that overexpression of syntenin increased the ABCB1 and also the CD44 at 1 day specifically at mRNA, where we were observing more clearly the results. So next, we think in inhibit SARC by another method, not by genetically, but pharmacologically, using the standard inhibitor of SARC, that is dasatinib. So the inhibition of SARC by dasatinib, we observe that the inhibition is at the level of the phosphorylation of SARC. This inhibition also inhibits the expression of CD44 and lightly in SOX2. So then we combine both drugs, the cisplatin and the dasatinib, to see if we could sensitize this highly metastatic cell line. So indeed, the combination of both drugs at different concentration was able to reduce this sensitization of this cell, specifically in the resistant cells that was highly resistant to cisplatin. No? So the combination of both drugs has a synergistic effect. Finally, we decide to study in vivo the inhibition of syntenin, as we observed that the inhibition was quite constant during the time, for more than 18 days in this particular laryngeal cell line. We decided to do the experiment by means of CIRNAs. We verify and monitor the animals during up to 44 days post-injection. And we observe that in all cases, in parental cells and in resistant cells, the effect of the sirna was clearly compromising the proliferation of the tumor. Interestingly, we observed that the number of mitosis decreased very significantly in the depleted cells versus the control in parental and resistant cells. And also the syntenin was decreasing the number of cells positive for the proliferative marker Ki67. Another interesting issue from the in vivo experiment is that we observed that the resistant cell, as we can see by the pictures, the resistant cells in comparison with the parental cells, in both cases, I'm talking about the control, they exhibit a fusocellular pattern that is quite different from the parental cells that are quite exhibit a epithelial morphology. So in the case of these resistant cells, the inhibition of syntenin was able to reverse this phenotype to a more epithelial-like as the parental cells. We observed only this in the case of the resistant, that was the only one that showed this fusocellular pattern. We wanted to verify if EMT transition was occurring in this resistant cell and if this EMT transition was reverted by the inhibition of syntenin. So for this purpose, we analyzed the relative mRNA levels of some stem cell genes and also N-cadherin, E-cadherin, SLAG, or TWIS, and other potential genes involved in EMT. So we observed that in vivo in these mice tumors, although we did not observe change in E-cadherin, we observed a decrease in N-cadherin in the sirna depleted cells for the syntenin 1, and also a decrease in the SLAG in the case of the resistant cells. Moreover, in both parental and resistant cell, we observe the decrease in the ABCB1 drug proliferative marker. Importantly, we observed that also the syntenin was able to decrease the metastatic ability of those cells in different organs. So we observed that in kidney and liver, the inhibition of syntenin compromise more specifically the parental cells. So when we analyze under the microscope the tumors formed in these mice, we observe the mitotic index that I was showing you in the graphs. So in the case of the syntenin parental cells, we did not observe any mitosis, and in the resistant cells, we did not observe hardly any in comparison with the control resistant, sorry. And for the Ki67, we observe also a considerable decrease in the cells positive for this proliferative marker. So indeed, syntenin was playing an important role compromising this proliferation of these tumors in vivo. This is some pictures of the metastatic organs for the presence of the cells inhibited by syntenin. In all cases, the signals are less than in the control cells. Then we did some kind of search by the in silico databases, by the TCGA, and we observed in cases when we compare normal and tumor tissue, in the primary tumor, this is the red box, was an overexpression of syntenin, with very significantly. And also the expression of syntenin was compromising the overall survivals in a group of head and neck cancers. So then we decide to analyze a big cohort of nearly 400 patients for the expression of synthenin and some of the proteins involved in EMT and also stemness. So by tissue microarrays, we were able to analyze a cohort of 382 head and neck squamous cell carcinoma patients. We were surprised because synthenin was not only located at the level of the cytoplasm, but also a minor proportion was detected in the membrane. This was a very few proportion of patients. I remember 3 out of the whole series, but a considerable number were expressing synthenin at nuclear levels in the nucleus of the cells. Then we studied another stem cell gene as NANOC, P21, ecadherin, Ki67, and Notch1. So the summary of all the immunohistochemistry study is given by this table. Before going to the numbers, I want to remark that we corroborated by another technique, in this case by Western blot, the overexpression of synthenin in a group of patients, and we corroborate the same results that we observed before with the immunohistochemistry technique. So indeed, synthenin was overexpressed in a proportion of patients. And now we go to the numbers. At cytoplasmic level, we observe 44%. At nuclear level, half of the patients, 99%, very few of the membrane. And then the expression of the other proteins, SARC, 63%, P21, 62%, very highly proportion of patients with NANOC, ecadherin, 55%. We assign 2 different levels of Ki67, high 12% and high and moderate 76%, and also the presence of Notch. So when we correlate and all the analysis and all the statistical studies, we observe that the presence of cytoplasmic synthenin was strongly correlated and very significantly with the presence of active SARC, the phosphorylation form of SARC, inversely correlated with ecadherin and P21. And the same results were also observed for the presence of cytoplasmic synthenin. So in relation with, for the nuclear synthenin, I think they are not included in the slide. Also, we observed direct correlation with NANOC, Ki67, and inverse correlation with Notch nuclear. Importantly, the expression of synthenin was correlated as well with tumor stage. As the tumor evolves through late stage, as 4 stage, the expression of synthenin at protein level increase. And also it increased as well very significantly with the tumor grade. As more aggressive and most heterogeneous is the tumor, most expression of synthenin 1. So apart from this, we observe a clear correlation as well with disease-free survival and overall survival. So in both cases, the expression of synthenin was compromising survival, and patients died before than those that did not express the synthenin. So we designed a model about what is the role of synthenin. Synthenin is a protein that is located in the cytoplasm membrane. Within that, in head and neck cancer, synthenin interacts possibly directly with SARC and activates this kinase by phosphorylation. and then at the same time is able to act through a panel of genes that are located at nuclear level, including in vivo, a panel of genes more directly related with metastasis as encadering or SLAC. At the same time, is also compromising the stemness of the cells by modulating ABCB1, CD44, and SOX2. Both are mRNA level and are protein level, and also somehow is involved in proliferation and apoptosis, because cells depleted for synthenin, they die by apoptosis. So the conclusions of the study is that first, resistant cells and cancer-resistant cells represents the main determinants of therapy resistance and recurrence. And synthenin is involved in chemoresistance, stemness, and metastasis. SRC inhibition, which according with our experiment is a direct downstream target of synthenin, is able to reduce resistance and ABCB1 expression. Synthenin depletion led to a reduction in cancer-resistant cell-related genes, CD44, SOX2, NANOC, OCT34, CD133, KLF4, and ABCB1. This reduction was particularly remarkable in cisplatin-resistant cells. In vivo, mice tumors formed by synthenin-depleted cells were smaller in size, harbor a lower mitotic index, and Ki67 positivity than those formed by control cells. Moreover, synthenin depletion reversed the fusocellular pattern exhibited by laryngeal resistant cells to a more epithelial phenotype. Synthenin expression was associated with the expression of active SAR, NANOC, and Ki67, and inversely associated with icatharin, P21, and Notch1. Lastly, synthenin expression was associated with SAR activation, poor differentiated tumor grade, advanced tumor stage, and shorter survival rates. So this obviously has a great impact in prognosis and poor clinical outcomes. The implications in the clinics. Since the use of SRC or ABCB1 inhibitors has shown modest results in the clinic due to their high toxicity, the targeting of synthenin could be a more effective therapeutic strategy to eliminate resistant cells in those head and neck patients as a effective way, no? So the challenge ahead is at our laboratory now is the search for novel drugs against synthenin 1. And just in science, enthusiasm, perseverance, and hard work, además, enjoy the run. And lastly, I want to acknowledge to all members of my laboratory and all collaborators here and the hospital and also abroad. Thank you very much.
Thank you, Matilde, for this elegant talk and very interesting data. Are there any questions from the audience? If not, I have a question. Have you related your data, or the clinical data on synthenin with the HPV virus status and with the EGFR?
No, we have not done that because, first of all, regarding the epidermal growth factor receptor, is intriguing why in, let's say, 95% of patients with head and neck cancer, they have overexpression of this protein, but somehow has no evidence in the therapeutic, as opposite of other cancer types, that the overexpression of this protein has really a specific target therapy, but is not the case for this patient. So if really more than 95% overexpress this, has not a lot of maybe, no.
But perhaps this is the resistance mechanism. So
yeah,
it might be a kind of pathway, bypass pathway.
No, this is true. And regarding the papillomaviruses, we know that also the percentage, at least here in Spain, is quite high. Of the distribution in Europe is very different regarding the influence for this virus in this head and neck cancer type. But here, at least in Spain, it's quite high. So it is remarkable that tumors that or patients that are positive for this virus has best prognosis, no? So we have not done that. We may explore this in the future. Thank you.
Okay, thank you. Okay, so we have to move to the next session, to the next talk. So thank you, Matilde, for your talk. And the next person will be the Stefania Schiattano, if I pronounced it right, from the University Magna Grazia from Catanzaro, from Italy. Please, the screen is yours.
Yeah, can you hear me?
Yes, we can hear you.
Okay. So, good evening, and thanks to the organizer for the...
Stefania, could you enlarge? So could you move to the full screen?
Okay. It's okay?
Yeah, now it works. Thank you.
Okay. So good evening. Thank you for this opportunity to talk about our work that regards the zinc finger 521 and its cooperation with the sonic edge pathway in the regulation of medulloblastomas. Zinc finger protein is a transcriptional cofactor containing 30 zinc finger, and that was identified for its selectively and abundant expression in early hematopoietic progenitors. ZNF1 has contained an N-terminal motif of 12 amino acids that is able to bind the nerve complex, the nucleosome remodeling, and histone deacetasis complex. And the deletion of this motif abolishes the binding to the nerve complex components. So ZNF1 is expressed in several human tissue and abundant also in brain. And among all the brain region is highly expressed in cerebellum, and in particular in the external granular layer of the cerebellum that hosts the granular neural precursor cells that are considered the cells of origin of medulloblastomas tumor. ZNF1 or ZFP1, the neurin counterpart of human ZNF1, has also identified as a muscle regulator during neural differentiation. So medulloblastoma are the most common high-grade childhood brain tumors, accounting 15–20% of all childhood brain tumors. And it's divided in distinct molecular subgroups characterized on their specific gene expression profile. So we analyzed in 436 cases of medulloblastomas compared to 18 normal cerebellum adult specimens and fetal specimens, the expression levels of ZNF1, and observes that in two particular groups, the group 4 characterized by NICN and the group 2 characterized by the dysregulation activity of sonic edge pathway, ZNF is particularly abundant. So, to test if ZNF1 has a role in the regulation of human medulloblastomas, we use DAOI human medulloblastoma cells that derived from a biopsy of desmoplastic medulloblastoma. The intermediate expression, both in terms of mRNA and protein of ZNF1 in the cells, consent to investigate also gain of function and the loss of function of this cofactor. So in gain of function studies, we use lentiviral vectors containing the cDNA for the full length of ZNF1 and for its deletion mutant. the cDNA deleted of the nerve binding domain of ZNF. In the loss of function studies, we utilize two different shRNAs for ZNF51. First of all, we observe the growth in other conditions of the transducer cells by MTS and cell counts, and observed that in both the experiment, ZNF51 overexpressing cells demonstrate a higher growth rate compared to its deletion mutant and the control vector. And when the cells were grown as spheroid in anchorage dependent conditions, the culture overexpressing ZNF51 had higher total number of cells and of sphere, and former also larger spheres. So to test the clonogenic potential of the cells, we performed also a single cell adherent culture and soft agar colonies assay. And in both the assay, ZNF51 demonstrate to enhance and over twofold the clonogenic potential of the cells. Conversely, the silencing of ZNF51 resulted in a maybe threefold decrease in the clonogenicity limiting dilution assay. We evaluate also the migration potential of those cells by Windling assay. And as shown, the OI expressing ZNF display a much higher migration across the scratch, whose width was reduced by approximately 15% in 6 hours, and almost completely covered by 24 hours. In contrast, the scratch was still evident after 24 hours in culture transduced with the deletion mutant or ZNF or the void vector. So, to test if the overexpression of ZNF51 is also associated with increased in vivo tumorigenicity in the whole cells, we carried a xenograft study. The initial experiment was performed to establish which cell dosage would be enabled to generate tumor in 100% of recipient mice. And so we utilize 3 different concentrations, two high dose and 1 low dose of cells for mice. So the result was that in all this experiment, tumors were observed in all animals inoculated with ZNF DAOI overexpressing ZNF51, irrespective of the number of cells. In contrast, 3 mice in each cohort inoculated with high doses of VHIR, the void vector, and only 1 mice in the group injected with low dose developed the detected tumors. So, in further experiment, a larger cohort of 8 and 5 mice were inoculated with low doses of cells per mouse, and as shown, the cells expressing ZNF51 demonstrate in both the experiments a much higher tumorigenic potential than those transduced with the deletion mutant for ZNF or the void vector. The tissue of tumors analyzed display similar histology. The difference is immunohistochemical. When the tissue were analyzed with an anti-ZNF51 antibody, demonstrated that the presence of high levels of the protein in all cells in the tumor deriving from DAOI ZNF51 full length or ZNF deletion mutant transducer cells compared to those generated by control cells expressing only the endogenous protein. So, to gain further evidence of the involvement of ZNF521 in the pathogenesis of medulloblastoma, we investigate the role of ZFP521 in a mouse model using mice with haploinsufficiency of the patched genes that encodes the regulatory subunit of the hedgehog receptor complex. So these mice develop cerebellar tumors that can recapitulate the feature of human medulloblastomas. The abundant expression of ZFP521 in these tumors is consistent with the high levels of ZNF521 expression observed in the sonic hedgehog subgroup of human medulloblastomas. So we decided to silence this abundant expression with two different shRNAs. The growth of the cells were isolated and grow under anchorage condition, and the growth was assisted by BRD incorporation. As documented, the proliferation rate of cells where ZFP521 expression had been silenced was significantly reduced. So we then asked if ZFP521 were required for or contribute to the tumorigenicity of patched medulloblastoma cells. And to this sense, we injected these cells in CD1 and nude mice. The tumor growth was monitored, and the results demonstrate a considerable reduction in tumor initiation and average size in the mice injected with ZFP521 silenced cells compared to the controls. So we can conclude that ZNF521 is a relevant regulatory factor in medulloblastoma. Our data implicated the stem cell and progenitor cell factor ZNF521 and also its association with the NERD complex in the control of the growth of at least a subset of medulloblastomas. The effect of ZNF on the chronogenic and in vivo growth of human and murine medulloblastoma cells suggests a potential requirement in stem-like tumor cells. So, we saw that ZNF521 is abundantly expressed in the sonic hedgehog subgroup of human medulloblastoma. So, in additional analysis of a larger cohort of patients, we observe a striking association between the transcript of ZNF521 with those of GLI1, GLI2, and PACHD1 that are the major effector of the hedgehog pathway. And we decided to study the possible crosstalk that can occur between ZNF and hedgehog signaling. So, as shown, the presence of ZNF521 enhances of twofold the expression of GLI1 and patched mRNA in the OI cells, and this effect was reinforced by the treatment with the smoothened agonist SAD. And this also value for the protein at the protein level. Okay. We next analyzed how ZNF521 modulates hedgehog signaling and the overall transcriptome of the OI cells by RNA-seq, and we observed that the overexpression of ZNF521 modulates 954 genes, while the SAG treatment of these cells modulates 892 genes. Interesting, when we overlap these two sets, we found 254 genes that were modulated, and we found that the top scoring pathway between these genes were related to the sonic hedgehog mechanism. to test whether a physical interaction occurred between ZNF and the G factor, we performed several IPs where we could transfect the HA tagged ZNF521 with the flag tagged Gli1 or Gli2, and immunoprecipitate the extract with the flag antibody, and the results showed the presence of Gli1 and Gli2 and the co-immunoprecipitation of ZNF521 with these with the Gli factors. Vice versa, if we immunoprecipitate with anti-HA antibodies ZNF521, we can also co-immunoprecipitate Gli1 and Gli2. To test if this interaction occurs at chromatin levels, we perform also a chromatin immunoprecipitation assay, where we transfect cells with Gli1 flag, ZNF521 tagged flag, and co-transfect flag tagged ZNF plus 3 per HA Gli1. The shared DNA pulled down with anti-flag antibody or with immunoglobulin G as control. We analyzed the presence of Gli1 binding sites by amplifying across the site with specific primers. So this assay showed that both Gli1 and ZNF521 were each pulled down in correspondence to Gli binding site consensus region. This was found both for Gli1 and patched 1 promoter, but not for distal sites or of Gli1 promoter, where no Gli binding sites are present, and we use as control. To assist the contribution of ZNF in the modulation of the activity of Gli factors, we performed also transactivation assay, where we use a reporter construct containing Gli binding sites fused to the cDNA of luciferase. And to this end, we could respect the patched reporter, 8 per Gli reporter and 12 per Gli reporter, that respectively as 1, 8, and 12 Gli binding sites. We contrast the reporter with Gli1 or Gli2 and with the increasing amount of ZNF521. As expected, both Gli1 and Gli2 alone enhance, transactivate the reporter activity. Instead, the co-expression of ZNF521 with the Gli proteins significantly enhance the transactivation of the reporter in a dose-dependent manner. This phenomenon is particularly striking with the 8 per Gli luciferases, where we have a superinduction up to 10 to 30-fold greater than that induced by Gli1 or Gli2 alone. And we also demonstrate that this superinduction is sensitive to the Gli antagonist GANT61. To test if the interaction between the nerve binding domain of ZNF521 and the nerve complex is relevant in this cooperation between ZNF521 and adjuvant signaling, we performed experiment in which we co-transfected the constant amount of flag DDK Gli1 with increasing amount of flag of HA tagged ZNF521, and vice versa, co-transfected the increasing amount of HA Gli1 with the constant amount of flag tagged ZNF521, sorry. The extract were immunoprecipitated by an anti-flag DDK antibody, and the immunoprecipitate analyzed by ZNF521, Gli1, and HDAC1 expression. The result, as expected, we saw that ZNF-FT1 co-immunoprecipitate with Gli1 and that Gli1 co-immunoprecipitate with ZNF-FT1. But the new data, the importance of these EPs, is that is the presence of the detectable co-immunoprecipitation of endogenous HDAC that increase proportionally with increased amount of ZNF-FT1, but not with increasing amount of Gli1. We also demonstrate this data were also confirmed by the measurement of HDAC activity. And we also demonstrate that the deletion mutant of ZNF-FT1 results in lower enhancement of the H-per-V luciferase reporter activities than that of the full length for ZNF-FT1 and Gli1. And also that this transactivation was sensitive to the treatment with the HDAC inhibitors, tricostatin A, sodium butyrate, and valproic acid. The same results were confirmed also for Gli2, and taken together, we can say that the data described above indicate that the association between ZNF-FT1 and the HDAC nerve complex takes part in the transcriptional synergism between ZNF-FT1 and HDAC pathway factors. So we have previously shown that the stem cell associated factor ZNF-FT1 whose expression is abundant in medulloblastomas of the sonic hedgehog subgroup enhance the growth, the chronogenicity, the motility, and the tumorigenicity of human medulloblastoma cells, and that ZNF-FT1 is required for the growth and tumorigenicity of mouse patched medulloblastoma cells. And now we can say that ZNF-FT1 has a potent hedgehog agonist whose mechanism of action depends on its interaction with the nerve complex. And this result is interesting because it identifies ZNF as agonist of hedgehog, and this crosstalk between ZNF and hedgehog may be considered a relevance in several types of tumors where hedgehog contributes to control the homeostasis of cancer stem cell compartments, and the ZNF is maybe highly expressed. So I want to thank all my collaborators in this project and all of you for your attention. Thank you.
Thank you, Stefania, for interesting study and interesting talk. Are there any questions?
Okay.
Do you think that this mechanism might be universal for other tumor entities? This is what you mentioned in the end of your talk. So, are there any tumor entities where it is important or it's studied?
Yeah, we know that the hedgehog signal is involved in several tumors, not also in medulloblastoma, but for example also in ovarian carcinoma. Human ovarian carcinoma, and that regulates the stem cell compartments of these cells. Actually, several trials with hedgehog inhibitors as vismodegib are used to treat the patients. So we think that if a real cooperation between ZNF-FT1 and hedgehog exists, this may be of potent use for the treatment of the patients.
Mm-hmm. And do you expect that there might be also inhibitors for zinc finger proteins
okay.
For humans or?
For zinc finger protein, actually there are no one inhibitors, but there are several inhibitors that we are trying for the nerve complex to inhibit the binding to the nerve complex of ZNF-FT1, and so to inhibit the enhancement of tumorigenicity in the of cells.
Okay, thank you.
Thank you.
Okay, and now the last talk in this afternoon session, and we invite Chloe Prunier from Innovation from France. I see that Chloe is already there, and
Yes.
please, the screen is yours.
Okay.
We are looking forward.
Thank you. Okay, good afternoon, everyone. So first, I'd like to thank the organizing team for giving me this opportunity to talk today. I'm Chloé Prunier. I'm a research project manager at Innovation. We are a French contract research organization offering a unique in vivo platform for oncology, the CAM-assay, that carry out human cancer xenografts in chicken embryos. So today I'm going to discuss about Innovation CAM-assay and its opportunities in the preclinical development of antimetastatic drugs.
Sorry, Chloe, can you
Yes.
enlarge the presentation? Can you put it into full screen?
I'm not in full screen right now.
And no, not
No?
yet. No.
Okay, so let me see, because I am on full screen on mine, so it's strange. Let's do it again.
Now it works. Thank
Now
you.
it works? Okay.
Yeah,
great.
now it works fine.
Okay. Yeah, so first I want to give a short overview of the antimetastatic drugs market, because even though there is a clinical urge to develop antimetastatic drugs, there is roughly 10 products available and approved in the clinic nowadays. Those drugs include vaccines such as Provenge, immunotherapies, targeted therapies, or even chemotherapies. But all of them were not initially discovered for their metastatic targeting activities, but for more anti-proliferative activities. In addition, there is roughly 60 molecules that are in clinical development, but did not reach the market yet. So those numbers, I think, prove that the development pipeline for antimetastatic drugs is not as efficient as it should and need to be adjusted. Indeed, several past efforts to target the metastatic process have resulted in failure. This is the case for MMPs inhibitor, SRC inhibitor, or even anti-VEGFR antibody. And all of them suffer from a lack of consistency between preclinical model and the clinical trials. But hopefully mistakes from the past were clearly identified, and in the last few years, efforts have been made to improve clinical trials and guidance to sponsors. For instance, working group, including academic, industry, and regulatory bodies were created to address those critical issues. For instance, in 2018, a new human endpoint called metastasis-free survival was approved by the FDA and allow apalutamide and darolutamide to be used for this purpose in prostate cancer patients. Additionally, the FDA emitted a guidance repository for sponsors who design a clinical trial aiming to find therapy against brain metastasis from solid cancer. So there are really been some improvement. And as I said, so preclinical models that recapitulates the different steps of the metastatic cascade and reflects the pathogenesis in human are essential to improve antimetastatic drug development. Mouse models have some limitation that I'm not going to discuss right now. And the chicken embryo that I'm going to introduce is an interesting alternative as it recapitulates all the different steps of the metastatic cascade in a short period of time. It is a fast, reliable, and affordable model that I will now introduce. So the CAM assay for chicken embryo chorioallantoic membrane is used as a platform to evaluate drug efficacy and toxicity in the oncology field. The CAM is a highly vascularized membrane that is used that allow efficient gas exchange through the shell, and it confers a respiratory-like function. It can be assimilated to the placenta in mammals. So it contains extracellular matrix proteins such as fibronectin, laminin, collagen type 1, and integrin. And due to its characteristics, the CAM is a good soil for tumor to grow. As you can see on the scheme on the left, at innovation, cancer cells are grafted on the upper part of the CAM at 9 days of embryonic development, or in short, E9. And over a period of 9 days, so from E9 to E18, cancer cells will form a tumor. They will remodel their microenvironment, and if they are invasive, they will spread to a distant tissue. So from 24 hours after the graft, from E10, it is possible to administrate therapeutic compounds such as reference compounds, standard of cares, or experimental compounds. And the efficacy and toxicity of a compounds is always evaluated alongside a control group that is treated with the vehicle alone. So the CAM assay allows to measure different aspects of cancer progression, such as tumor growth, metastatic invasion, angiogenesis, immune cells infiltration, and many others. The CAM assay is a complete in vivo model. Administration of compound is made during the second part of the embryo development, and at that stage, all organs are present and functional. This is why we call it an adult embryo. Moreover, the embryo metabolism and immune system are active. The thymus, the bursa of Fabricius, and the spleen are formed. Moreover, the different population of immune cells can be identified in the peripheral blood, as shown on this figure. And finally, infiltration of T cells, dendritic cells, NK cells, and macrophages within the tumor can be observed. So if you are interested in learning more about the immune system of the chicken embryo, I encourage you to read our latest review published in VHIR in Immunology. Moreover, tumors grown on the CAM have a histologic profile that is similar to that obtained in mouse models and in humans. This has been confirmed with several cell lines, breast cancer, melanoma, lung, among others. And there is a real tumor microenvironment with fibroblast infiltration, blood vessels, and as I said, immune cells infiltration sometimes. So implementing the CAM assay to different models of cancer is not trivial and requires expertise. At Innovation, we have spent more than 5 years to standardize the CAM assay for diverse cancer. And we now have very low standard deviation and very low p-value as well. And the dose response is also very good. We have developed several standard of care for every model, and we have very good dose response. Lastly, we wanted to make sure that our model conserve the feature of the human cancer cell line. So we use transcriptomic to look at a 10,000 genes library and compare expression of this library between cell line culture in vitro and xenograft in ovo. And we saw that there was a very good correlation between both conditions. And the few variations observed were really related to the tumor microenvironment, epithelial to mesenchymal transition, and also modification of the extracellular matrix, which is very consistent when you switch from a 2D to a 3D environment. So to summarize, the CAM assay is an attractive preclinical model as it is fast. You have results within a month. It is very sensitive. You need less compound to perform your study. It is reliable as we have standardized it and we have a high statistical power, and it's affordable. It's also a validated model for immuno-oncology and especially checkpoints inhibitor on TPD1, PD-L1 agent. Moreover, it's an alternative to animal experimentation, and it is not subjected to 3R constraints. And this is an important point, especially nowadays. Finally, there is an excellent predictivity with respect to mouse test results. There is 100% correlation for reference molecules. Okay, so I'm now moving on to the next part of my talk that is related to the experimental setup and the readout that are available with the CAM assay. At Innovation, we use the CAM assay to answer a large panel of scientific questions as we provide a large range of study possibilities. In terms of compound types, we have experience with small molecules up to viruses. We have covered so far a large panel of cell line, including PDX and CTCs. Treatment types are various and goes from synergistic study to radiation. And finally, we have developed a large catalog of readouts that we can perform, and I will present some of them in the next slides. But before, I just want to stress that we have standardized the CAM assay for a large range of cancer, approximately 50 classical human cancer cell line that represent nearly all types of cancer. So if you can't find your usual suspect, sorry, in this list, we are setting up new model approximately every month. So it's really doable. Okay, so regarding early toxicity assessment, we provide a death ratio index that corresponds to the relative percentage of eggs that are alive or dead at the end of the experiment. A Kaplan-Meier curve is also provided to illustrate the dynamic survival during the experiment. If you need a deeper analysis of the toxicity, it is possible to do that through 22 body checkpoint abnormalities checkpoints, or also to perform histological analysis on various tissue or organ that are specific to your research question. Regarding tumor growth, tumors are collected at the end of the experiment and weighted to provide a tumor regression index. This can be done to test a drug efficacy, but also to test the tumorigenesis capacity of a modified cell line. We can also take some picture to illustrate the tumor. Of course, you can perform, well, we can perform, or you can also do it, but perform other kind of analysis on those collected tumors, such as histology analysis, proteomics, immune cells infiltration, and others. So metastatic invasion is observed in novo. It has been shown in the literature. So if cells are invasive, they will detach from the primary tumor and colonize distant tissues, so the lower part of the CAM, but also they can reach organ from the embryo. So it's well established that all the steps of the metastatic cascade are recapitulated in the chicken embryo. Using microscopy technique, researchers have shown that the early dissemination is happening in the surrounding tumor stroma. Intravitreal microscopy experiments allow to monitor the invasive front of the tumor and to measure migration speed, displacement, and directionality of cells. CTCs were also detected in the bloodstream and isolated. Distant metastases at various sites were identified using PET scan imaging, bioluminescence, CT scan, or even whole body fluorescence emission. So altogether, I think it really shows that this assay is very relevant, and it is a spontaneous metastasis assay that allows metastasis evaluation in a 9 days experimental period. At Innovation, we have chosen a very fast and sensitive detection technique to measure the presence of metastatic foci. So we perform real-time qPCR to measure the presence of a specific allele sequence to detect human cells in chicken tissue. In routine, we mainly analyze the lower part of the CAM and provide a relative amount of metastasis compared to the control group. However, it's possible to perform the analysis in other organs, such as the brain, the liver, or the lung, upon request. Let's now move to some case study to better appreciate the CAM assay usefulness for anti-metastatic drug screening. First, I want to share some information regarding metastasis tropism. So we investigated this aspect in the invasive MDA MB231 breast cancer model. We grafted cells on top of the CAM, and 18 days after, we collected the lower part of the CAM at the opposite side of the grafting site. And we look at the presence of allele sequence in different organs. And we observed that there was a preference for cells to invade liver, lung, and bone that is consistent with what is observed in the clinic. We also observed that the lower part of the CAM was a site of choice for cells to invade compared to the embryo organ. And finally, we could detect a lot of circulating tumor cells in this MDA-MB231 model. So you can see here that the amount is really huge compared to what we can find in the lower part of the CAM. A second case study that presents the antimetastatic activity of compound A in a non-small cell lung cancer A549 model. So 18 days after grafting cells on top of the tumor, the lower part of the CAM was collected and the presence of allele sequence detected. So whereas compound A does not affect tumor growth at all, we observe that it significantly decreased the amount of metastasis in the lower part of the CAM, suggesting that this compound has really specific antimetastatic activity. Another case study presents the antimetastatic activity of several compounds, again on non-small cell lung cancer A549 model. Cells were grafted on top of the CAM, and 18 days after, first tumor were weighted, and you have here the result. We can see that only the standard of care number 2 induced an antitumoral effect. But then we look at metastasis in the brain, and interestingly, standard of care number 2 did not impact the metastatic load at all, but standard of care 1, compound A, compound B, and compound D did really decrease significantly the metastatic load, suggesting that those compounds have, again, a very specific antimetastatic properties. So here is another case study that is related to target validation using engineered cell line. So the investigation was focused on the role of the RAPTOR and the mTOR pathway in the epithelial maintenance to prevent metastasis. So you know that epithelial to mesenchymal transition is happening in the early steps of the metastatic cascade, and cancer cells have to switch from this epithelial phenotype to a mesenchymal one in order to move in the surrounding stroma and reach the blood vessel. So in this paper, Michaelian and colleague used our CAM-assay to look whether inhibition of Raptor or mTOR pathway boosted EMT and potentially lead to metastatic invasion. And to do so, MCF10A cells were grafted on the upper part of the CAM, and either genetic modification of Raptor or pharmacological inhibition of mTOR was performed to control protein expression and activity. And the analysis of the lower part of the CAM at the end of the experiment revealed that both genetic and chemical inhibition increased the number of nodules when either mTOR or Raptor was inhibited. So really suggesting that they indeed have a role in this epithelial maintenance. Lastly, a case study that illustrates correlation between results obtained in the CAM assay and in the mouse. In this paper, Cross and colleague used our CAM assay to look whether overexpression of a small miRNA 135A was involved in metastatic spreading of prostate cancer cell, PC3. So either wild-type PC3 or PC3 modified to overexpress mRNA 135A were grafted on top of the CAM, and 18 days after, the number of foci was counted in the lower part of the CAM. And we observed at that time that overexpression of the mRNA led to nodule regression without affecting tumor growth at all. So these results were confirmed a year later in the mouse and proved that there is consistency between what we observed at that time and what is observed in the mouse. To finish, I present you the list of our customer publication or patents, just to show you that this model is well recognized by reviewers. And we are always here to support our customer in the writing of future manuscript and helping in the Innovo related part, and also to answer reviewers' question. So I hope I convinced you that the chicken embryo model is a relevant in vivo model that can help you save some time and money in your drug development journey. Today, we focus on anti-metastatic drug development. The CAM assay is a well-established model for metastasis analysis, and we use it at Innovation to identify a potential anti-metastatic drug, but also to validate a target involved in the metastatic cascade. So we maximize the probability of success and reduce time and costs of your preclinical study. We are dedicated to improve R&D productivity and drug discovery, decrease and circumscribe risk in preclinical phases, and help our customer to make solid go-no-go decision. Please feel free to contact us to discuss your in vivo assay and drive your research forward. As a research company, we are always flexible and available to address your need. Thank you very much for your attention. I'll be happy to take some questions.
Thank you very much for this presentation. I see that there is one question in the chat. Perhaps I should read it. Very nice presentation. Do you see signs of immune rejection at later time points of your assay? How well your assay works with PDXs?
So it can happen with some cell line that indeed tumor grow and up to a point where they start to not grow anymore, and even sometimes to regress. But this is very rare. But I think this might be indeed an information that there could be some immune rejection at some point. Because as I told you, the immune system start to be mature at 10.5 days of embryonic development. So from this moment on, we have mature T lymphocytes, we have dendritic cells, NK cells. But this is very rare, I have to say.
And what is the lowest tumor cells load which you have to apply in this assay? How many cells?
In terms of concentration. So for cell line, we are always between 0.5 and 3 million, I would say. But we have worked with CTCs, and because, you know, CTCs are very rare, we grafted very few, and I think it was below 500,000, so even 100 sometimes. But of course, the efficiency of the graft and of the growth is really reduced.
So for the CTC samples, so you use then the metastatic patients, because 100, 500 cells, this is very much for.
Yeah, I know. But the thing is, sometimes we pull them. For human patient material, we don't pull them. So, and here we are more between 100 and 50 and 500, I would say. But we did some experiment where we extracted CTCs from the embryo and we grafted them so we could pull them, and here we could graft more cells.
Okay, and which method do you use for CTC isolation to capture them from patients for this grafting?
So we have a collaboration with an hospital in Lyon that is in charge of this as isolation, and we don't perform it in-house. It's now not our expertise.
Okay, but you don't know if it is Parsortix or any other CTC system which is available.
Which one they use? I think it's fluidic. It's fluidic system.
Okay.
Microfluidic system, yeah.
And for CTCs, when you graft them, how large are the tumors after culturing them?
So it really depends. It's very small compared to what we can have with a cell line, but at least we can observe efficacy of drugs on it. So it's big enough so that we can perform efficacy study.
Okay.
I think maybe it might be around 10 milligrams or something. I should have my colleague that is in charge of this project. I'm not completely sure, but we can discuss if you're interested.
Yeah, sure. So if there are no other questions, thank you, Chloe, for your interesting talk. And I think there is a coffee break in the program right now, so enjoy it. And after this,